Okay, now we're going over to cell biology in Neet, which is equally or very much related to our studies of life sciences in the NET, CSIR NET. So cell biology here contains tools and techniques in cytology, cells as a unit of life, biomembranes, structural organization of cells, biomolecules, enzymes, cellular metabolism, and cell reproduction. So those of you NEET students, pay attention. I'm going over to the subject of biomolecules, enzymes, cellular metabolism. We're going to that page 188 here. In connection with what I've been explaining on the National Eligibility Test of the CSIR NET Life Sciences, regular 2, in relation to cellular metabolism, I'm giving you a continuation for the Neet Biology section with cellular metabolism in your Neet syllabus. So here we go into the subject of cellular metabolism, where metabolism is the biochemical modification of chemical compounds on living organisms and cells. So this is a more simpler version as compared to the Neet version, so it's easier for you to understand. And as you listen to my lecture, if you have any doubts, please give them in the comment section or ask questions, and I'll give you answers to your questions with explanations, okay, if any required. And it's through the process of metabolism that organisms process nutrients and the biochemical tools and structures they need to maintain a living state. Cell metabolism involves extreme complex sequences of controlled chemical reactions called metabolic pathways, usually a sequence of enzymatic steps. Enzymes are crucial to metabolism because they allow organisms to greatly accelerate slow, favorable reactions as well as couple unfavorable reactions to avail energy sources. By providing energy, usually in the form of ATP, to metabolic processes, cells can successfully power reactions that would otherwise never occur. So metabolism has two distinct divisions: catabolism, which is a destructive process, and anabolism, which is a constructive process. Now, catabolism is a type of metabolic process occurring in living cells by which complex molecules are broken down to produce energy and reducing power. Catabolic reactions are primarily oxidation reactions. The primary purpose of catabolism is to regenerate ATP, the primary energy currency of all cells on balance. Catabolic reactions are normally exothermic. Anabolism is a constructive metabolic process whereby energy is consumed to synthesize or combine simpler substances, such as amino acids, into more complex organic compounds, such as enzymes and nucleic acids. Anabolic reactions are build-up reactions, which are generally entagonic or energy-absorbing. Right? And carbohydrate metabolism. The major function of carbohydrate metabolism is as a fuel to be oxidized and to provide energy for other metabolic processes. The three principal monosaccharides absorbed from the food are glucose, fructose, and galactose. Now, carbohydrate metabolism denotes the various biochemical processes responsible for the formation, breakdown, and interconversion of carbohydrates in living organisms. Carbohydrate metabolism includes carbon fixation, whereby CO2 is reduced to carbohydrate. Glycolysis, the breakdown of the glucose molecule in order to obtain ATP. The Pentose Phosphate pathway, which acts in the conversion of hexoses into pentoses and in NADPH regeneration. Glycogenesis, the conversion of excess glucose into glycogen in order to prevent excessive osmotic pressure buildup inside the cell. Glycogenolysis, the breakdown of glycogen into glucose in order to provide a steady level of glucose supply for glucose-dependent tissues. And gluconeogenesis, de novo synthesis of glucose molecules from simple organic compounds, that is, formation of glucose or glycogen from non-carbohydrate sources like fats and proteins. The oxidation of pyrubic acid to acetyl-CoA, a necessary step before the production of glycolysis could enter into the citric acid cycle. Citric acid cycle, final common pathway for the oxidation of carbohydrate, fat, and proteins. Now, students of NEET, I have given a much broader, detailed explanation for the NET students. So you can go to the start of my previous lecture on this. Each of my lectures have been titled with avid descriptions, extended descriptions and titles for each topic that has been dealt with each day. Go to it, you can listen to it, note down the points. If you cannot follow the lecture due to the speed or the flow, you don't get the flow, please refer to this transcript. And even after that, if you have any doubts, please come to the comment section and write your query, and I'll give you explanations, right? So here we finish up this topic on cellular metabolism, the initial part of it, and then we're continuing to about dicarboxylic acid shuttle, additional pathway for regeneration of dicarboxylic acids, right? Okay, dicarboxylic acid shuttle, additional pathway for regeneration of dicarboxylic acids. The reactions of carbohydrate metabolism cannot take place without the presence of the B vitamins, which function as coenzymes. Right? And phosphorus, magnesium, iron, copper, manganese, zinc, and chromium, also known as cofactors. Carbon fixation. Carbon fixation is a process found in autotrophs, usually driven by photosynthesis. I've given avid details about this in the previous lectures about photosynthesis. Please refer to those lectures and the transcripts for all explanations, right? Whereby carbon dioxide is converted into organic compounds. In plants, there are 3 types of carbon fixation, being C3, plant that uses the carbon cycle for the initial steps, then incorporates CO2 into organic matter, forming a 3-carbon compound as the first stable intermediate. Most broadleaf plants and plants in the temperate zones are C3. C4 plant that prefaces the carbon cycle with reactions that incorporate CO2 into 4-carbon compound. C4 plants have a distinct leaf anatomy. This pathway is found mostly in hot regions with intense sunlight. Tropical grasses such as sugarcane and maize are C4 plants, but there are many broadleaf plants that are C4 because they prefer the C4 plants that preface the Calvin cycle. C3 plants that can use the Calvin cycle for initial steps but incorporate CO2 into organic matter. So that's a difference there. So this pathway of C3 or C4 plant that prefaces the Calvin cycle with reactions that incorporate CO2 into 4-carbon compound. C4 plants have a distinct life anatomy. So this pathway is found mostly in hot regions with intense sunlight. Tropical grasses such as sugarcane and maize are C4 plants. But there are many broadleaf plants that are C4. CAM plant, that is CAM plant that uses crystallization acid metabolism. Right? I think I've already explained this in the previous lecture for net students. Refer to that, please. As an adaptation for arid conditions, the carbon dioxide entering the stomata during the night is converted into organic acids, which release CO2 for the carbon cycle during the day when the stomata is closed. The jade plant, Crassula ovata, and cactus species are typical of CAM plants. Next, the glycolysis. The sequence of, this is a continuation from the net, right? The glycolysis, the sequence of reactions or pathway by which glucose is degraded anaerobically to form pyruvic acid is called glycolysis. Glycolysis occurs in all living organisms and in all types of respiration. Hence, James called it as co-respiration. This is also known as fundamental respiration. Sites of glycolysis is cytoplasm. Hence, it's called cytoplasmic matrix of a living cell. The reduction of the glucose during glycolysis produces compounds for the synthesis of protein, fats, and the nucleic acids. Most of the steps in the glycolysis are reversible. Glycolysis may be divided into 2 phases, a preparatory phase and an oxidative phase. In the preparatory phase, breakdown of glucose and low energy phosphorylation occurs and energy is used. In the oxidative phase, high energy phosphate bonds are formed and energy is stored. All reserve substances first undergo hydrolysis and are finally converted into glucose. In majority of the organisms, glucose acts as the respiratory substrate. In glycolysis, 1 molecule of glucose is converted into 2 molecules of pyruvic acid. The sequence of reactions in glycolysis was traced by Emden-Meyerhoff-Parnish EMP. Hence, glycolysis is also called the EMP pathway. Number of ATP used for phosphorylation of glucose in glycolysis is 2. That's first and third reactions. Number of ATP formed during glycolysis is 4, that is 2 ADP for each GAP. Net gain of ATP during glycolysis is 2, that is 4 minus 2 equal to 2. If fructose 6-phosphate is oxidized through glycolysis, net gain at ATP is 3, that is 4 minus 1 is equal to 3. Number of pi utilized during glycolysis is 2. Alright, we can go back to respiration to get more details on that. Next is the pentose phosphate pathway. Pentose phosphate pathway is an alternative method of aerobic respiration and which occurs in the cytoplasm of mature plant cells and accounting for 60% of total respiration in liver cells, and also occurs in adipose tissue and lactating mammary glands. In this pathway, for every 6 molecules of glucose, 1 molecule is completely oxidized into CO2 and reduced coenzymes or 5 are regenerated. The pentose pathway is also known as phosphogluconate pathway. Since this pathway metabolizes glucose 6-phosphate with reactions that bypass the reaction of glycolysis, it is also known as hexose monophosphate shunt, HMP shunt. And more details of this in the chapter on respiration in plants. Glycogenesis is the synthesis of glycogen in animals and starch in plants from glucose. The chief sites of the formation of glycogen are liver and muscle. Liver glycogen is mainly concerned with the maintenance of the blood glucose, but muscle glycogen acts as a readily available source of hexose unit for glycolysis within the muscle itself. And for the presence of glucose in blood is also called blood sugar. So 100 ml of blood contains 80 to 100 milligrams of glucose during fasting and 100 to 120 milligrams per 100 ml of blood 2 hours after meal postprandial. So glycogenesis responds in both hormonal and electrical control. Glycogenesis is stimulated by pancreatic hormone insulin, growth hormone of pituitary, and cortisol of adrenal cortex. Glycogenesis takes place by a series of chemical reactions, and these are: glucose is phosphorylated into glucose 6-phosphate by ADP in the presence of Mg2+, and under the influence of an enzyme hexokinase in the liver. I think I've explained this for the net syllabus, so it's easier understood now. So we have glucose 6-phosphate, which is then converted to glucose 1-phosphate, is in an isomerization reaction catalyzed by the enzyme phosphoglucomutase. And glucose 1-phosphate reacts with uridine triphosphate UDP to form the active uridine diphosphate glucose UDPG complex, and inorganic phosphate pyrophosphates PPI are released. This reaction is catalyzed by the enzyme UDPG pyrophosphorylase. The first carbon of the activated glucose of UDPG form glycosidic bond with the carbon 4 of a terminal glucose residue of glycogen. This reaction is catalyzed by the enzyme glycogen synthetase and uridine diphosphate. UDP is liberated. Hence, a pre-existing glycogen chain becomes elongated as successive 1,4 linkages occur. When the newly formed straight chain of glycogen has been lengthened to between 6 and 11 glucose residues, a second enzyme branching enzyme, amylo 1 and 4,1-trans-glucosidase acts on the glycogen. This enzyme transfers as a part of the 1,4 chain to a nearly chain forming 1,6 linkage, thus establishing a branch point in the molecule. Hence, under the combined action of glycogen synthetase and branching enzyme, the molecule of glycogen is assembled. Glycogenolysis. Glycogenolysis is the conversion of glycogen to glucose, which could occur several hours after a meal or overnight in the liver or in the absence of glucose 6-phosphate in the muscle to lactate. Or glycogenolysis is the catabolism of glycogen by removal of a glucose monomer and addition of phosphate to produce glucose 1-phosphate. This derivative of glucose is then converted to glucose 6-phosphate and intermediate in glycolysis. Glycogenolysis transpires in the muscle and liver tissue where glycogen is stored as a hormonal response to epinephrine, example, adrenergic stimulation, and a glucagon, a pancreatic peptide triggered by low blood pressure concentration. Liver hepatic cells can consume the glucose 6-phosphate in the glycolysis or remove the phosphate group during the enzyme glucose 6-phosphatase and release the free glucose into the bloodstream for uptake by other cells. So muscle cells will not release glucose but instead use the glucose 6-phosphate in glycolysis. Glycogenolysis occurs in the following way. There's a breakdown of glycogen, which is initiated by the action of the enzyme phosphorylase. In the presence of inorganic phosphate, the enzyme catalyzes, which catalyzes the removal of 1,4-glycosyl residues from the outermost chains of glycogen molecule until about 4 glucose molecule residues remain on either side of 1,6 branch. The hydrolytic splitting of 1,6 linkages requires the action of a specific G-branching enzyme, imelo-1 and 6-glucosidase. The combined action of phosphorylase and D-branching enzymes converts glycogen to glucose 1-phosphate. Glucose 1-phosphate is then converted into glucose 6-phosphate by the action of enzymes glucose 6-phosphatase in the liver. This is the final step in hepatic glycogenolysis, and this is manifested by a rise in level of blood glucose. When glycogenolysis occurs in muscles, the reactions terminate in the glucose 6-phosphate stage because the liver phosphatase enzyme is absent in muscle. Gluconeogenesis. This is the formation of glucose from non-carbohydrate sources such as certain amino acids, primarily alanine and glutamine, and the glycerol fraction of fats when carbohydrate intake is limited. Liver is the main site for gluconeogenesis except during starvation, period of fasting, or intense exercise, and when the kidney becomes important in the process. The majority of enzymes responsible for gluconeogenesis are found in cytoplasm. The exceptions are in mitochondrial pyruvate carboxylase and mitochondrial pyruvate carboxykinase, which is located in the mitochondria. The rate of gluconeogenesis is ultimately controlled by the action of a key enzyme, fructose 1,6-biphosphorylase, which is also regulated through single-strand structure by CAMP and its phosphorylation. Alright, now gluconeogenesis cannot be considered to be a reverse process of glycolysis, as the 3 reversible steps in glycolysis are bypassed in gluconeogenesis. This is done to ensure that glycolysis and gluconeogenesis do not operate at the same time in the cell, making it a futile cycle. Therefore, it is a reciprocal regulated between glycolysis and gluconeogenesis. Many regulations which inhibit glycolysis will activate gluconeogenesis in reverse. 3 reversible steps in glycolysis are phosphoenol pyruvic acid plus ADP in the presence of pyruvic kinase giving pyruvic acid plus ATP, fructose 6-phosphate plus ADP in the presence of phosphofructokinase gives fructose 1,6-biphosphate plus ADP, and glucose by ATP in the presence of hexokinase gives glucose 6-phosphate plus ADP, which I've all explained to you long back. It's a good repetition for you for the NEET students. It's a good take up from here. In gluconeogenesis, the 3 irreversible reactions of glycolysis are bypassed by the following steps. Phosphoenol pyruvate formed from pyruvate by oxaloacetate. First, pyruvic acid is carboxylated to oxaloacetic acid at the expense of an ATP. The enzyme pyruvic carboxylase present in the mitochondria contains a covalently attached prosthetic group biotin, which serves as a carrier of activated CO2. Pyruvic acid plus CO2 plus ADP plus H2O in the presence of acetyl-CoA and magnesium 2 plus oxalic gives oxaloacetate plus ADP plus pi and 2H plus. Right? So oxaloacetic acid, the product of pyruvic carboxylic reaction, does not diffuse readily from the mitochondria. To achieve this end, oxaloacetic acid is reduced to malic acid inside the mitochondria by an NADH-linked malic acid, right? Dehydrogenase. Malic acid then diffuses across the mitochondrial membrane into the cytosol, where it is reoxidized to oxaloacetic acid by an NAD plus linked cytoplasmic malic dehydrogenase. Oxaloacetic acid plus NADH plus H plus in the presence of mitochondrial malic dehydrogenase gives malic acid plus NAD plus mitochondrial malic acid. In the presence of diffusion through mitochondrial membrane gives malic acid in the cytoplasm. Malic acid plus NAD plus in the presence of cytoplasmic malic dehydrogenase gives oxaloacetic acid plus NADH and hydrogen. In the cytosol, oxaloacetic acid is simultaneously carboxylated, phosphorylated by a second enzyme, phosphoenol pyruvate carboxylase, at the expense of a second high-energy phosphate bond provided by GDP. Oxaloacetate plus GDP in the presence of magnesium gives phosphoenol pyruvic acid plus CO2 plus GDP. The carbon dioxide that was added to pyruvic acid by pyruvic carboxylate comes off in this step. Fructose 6-phosphate is formed from fructose 1,6-biphosphorylase. The hydrolysis of fructose 1,6 biphosphate, is catalyzed by a specific enzyme fructose 1,6 biphosphorase. This is a key enzyme for the conversion of pyruvic acid and triphosphate to glucose. During gluconeogenesis, it is present in liver, kidney, and striated muscle. Glucose is formed by hydrolysis of glucose 6-phosphate. The conversion of glucose 6-phosphate to glucose is catalyzed by another specific phosphatase called glucose 6-phosphatase. It is present in intestine, liver, and kidney, where it allows tissues to add glucose to the blood. The enzyme is absent from the muscle and adipose tissue. 6 high-energy phosphate bonds are used to synthesize 1 molecule of glucose from 2 molecules of pyruvic acid in gluconeogenesis, but only 2 ADP were generated in glycolysis in the conversion of glucose to pyruvate. And 2 pyruvic acid plus 4 ADP plus 4 GDP plus 2 NADH2 plus 2 H2O gives glucose plus 4 ADP plus 2 GDP plus 6 PI and 2 NADI. And during 2 NAD plus, that's during the gluconeogenesis, 1 molecule of ADP is utilized in the conversion of each pyruvic acid to oxaloacetic acid in the mitochondria. Another one during the phosphorylation of 3-phosphoglyceric acid to 1,3-diphosphoglyseric acid in the cytoplasm. In addition to 2 GDP more molecules of ADP are required in the synthesis of 1 mole of glucose from 2 moles of pyruvic acid. Gluconeogenesis, occurring in the liver and kidney, maintains the glucose level and the blood so that brain and muscle, which have a high demand for the glucose, can extract sufficient amount of this vital substance to meet their metabolic demands. We'll meet you tomorrow with the remainder of the portions starting from oxidation of pyruvic acid. Then that followed by citric acid cycle, right? And then we will be having dicarboxylic acid shuttle. Disorders due to carbohydrate metabolism, hereditary fructose intolerance. All right? Okay.